Hi, I'm Hanna Choi. I'm a senior at UMass Amherst studying biology, and I'm a research assistant in the Blanchard lab. And for this past year, I've been analyzing the lipidomic and metabolomic data within the soil composition of our plots from Harvard Forest. So a little background, these plots were treated with temperature 5 degrees above ambient since 1991, and our samples were extracted in 2017 for analysis. And there were 4 methods used to analyze this, which is why we call this a multi-omic approach. We use FTICR-MS extraction and analysis to understand the carbon compounds or the carbon molecules found in the soil. We also analyzed a metatranscriptomic analysis or data. We also used liquid chromatography mass spectrometry to identify lipids, and gas chromatography mass spectrometry to identify metabolites. So this 4-pronged approach sort of helped us with multiple perspectives in understanding the soil composition in 2017 after about 20 years of heat treatment. So the FTICR-MS data showed that about 16,814 unique carbons existed, with about 57% we were able to find a molecular formula for, and about 15% were hit to a database online called KEGG, which sort of helps us navigate the understanding of these carbon molecules. So the FTICR-MS data was significant and important because it showed us that there was a shift in abundance in both horizons. So specifically, the organic or upper layer horizon of the soil had a decrease in abundance by 13% when compared to the control. In comparison, the mineral layer had an increase in abundance by 17%. So this transformation of carbon molecules from the organic to mineral layer is interesting and warrants further discussion. But to continue, the FTICR-MS data also showed a decrease in unique molecules within both horizons compared to the control. So this decrease in diversity is disheartening for soil in general, because we would like to have an increase in abundance of unique compounds so that we have a sense of diversity for microbial robustness and survivability. So in addition to that, metatranscriptomic data showed about 100, almost 200 million different protein coding reads. So these reads were assigned to 1,483 taxonomic groups. And in comparison to the control, there was a different level of diversity by class within these taxonomic groups, which is interesting, especially in the fungi. But Will is the one that had analyzed this, so he discussed this more in our manuscript. I was behind the lipidomic and metabolomic data, and essentially the lipids and metabolites that I analyzed supplements Will's work with the metatranscriptomic data. And just like he had, I found a lowered level of abundance within the mineral and organic layer in terms of number of metabolites and lipids. So this makes sense because this lowered level of abundance in the carbons available leads to a lowered abundance in lipids and metabolites. And after statistical analysis of the lipids and metabolites, I found about 30 in each group to be statistically different within the heated plot versus a control. I'd like to say that the liquid chromatography mass spectrometry method used to extract the lipids is very limited and doesn't represent the entire soil composition. So the statistical analysis within the soil or within the lipids should be taken with a grain of salt. But nonetheless, I found that there was no difference in the chain length or degree of saturation within the heated versus the control. Within the metabolites, specific lipids or specific metabolites were interesting to me because they differed in abundance within the heated in interesting ways. So specifically, sulfurose is a cellulase inducer, and cellulase is an enzyme that breaks down cellulose, which is a recalcitrant carbon. So recalcitrant carbons are important in our discussion because they're difficult to break down naturally compared to labile carbons. So a soil microbe's ability to break down recalcitrant carbons is maybe beneficial in the future with an increase in this kind of carbon. So the increase in sulfurose might indicate this shift in adapting microbes, but we're unsure of whether or not this is happening. We would need to analyze in lab using a cell respiration test. Anyways, other metabolites that were interesting were the specific amino acids like glutamate and proline, which have been indicated to serve as solutes for osmolar regulation in bacterial cells. So we can't really conclusively say anything about whether osmolar regulation within bacteria are being affected due to these statistically significantly changed levels of glutamate and proline, but it warrants further investigation. And so does the investigation of the difference between the L enantiomers and the D enantiomers that were statistically significant in the samples. And even finally, one other metabolite that I found interesting was dehydroadenine, which is a modifier of proteins that acts in the process of antimicrobial or even anti-tumors in mammals or in animals. So I guess what I want to say is that metabolomic data from this sample sort of provided us with a set of metabolites to look into in the future for further analysis to broaden the discussion of what the soil is actually doing on a molecular level. So in conclusion, the multi-omic plus FTICR-MS data has indicated a shift in diversity as well as a decrease in diversity, along with a lower level of abundance when heated 5 degrees Celsius above ambient. So this is worrisome news long-term because this is the effect that global warming will have in all populations of soil or all types, not just terrestrial soil. So it is something to consider in an ecological sense. If you have any questions of this poster, my email is down below. And I want to close off by thanking Dr. Blanchard, my PI, for the last 2 years. I've learned a lot about omics and a lot about coding and very non-biology oriented things that I found interesting and unique. If you'd like to look more into this poster, I have it linked as well. And that's about it. If you have any questions, let me know. Have a good one.